up next, we have Dr. Jelena Kolic. I love your name, by the way. So she's going to be chatting to us essentially about insulin secretion, how we're all unique, how we respond to it differently, and how we can all tailor our diets to better suit our needs. So without further ado, let's get our cheering on for insulin and for Dr. Jelena Kolic, everybody.
So I would like to start off by thanking the organizers for inviting us here today. I'm here representing the Diabetes Research Group at UBC, as well as the newly formed Institute for Personalized and Therapeutic Nutrition. I'd like to start off the talk by apologizing on behalf of my postdoctoral advisor, Dr. James Johnson, who is supposed to be here today, but he's unfortunately not so great at planning a calendar. Great research scientist, but had this event scheduled for December instead of January. So I am here today on his behalf. And today we're going to be dividing this talk today in two specific parts. And in the first part, we're going to be talking about the difference between a causal correlation and a direct causation. And so we're going to be talking about some causal relationships between insulin resistance, and weight gain. And we're going to talk about how scientists can use engineered mouse models to clarify some of these relationships. And then the second part of the talk today, we're going to be talking about how insulin secretion may be different between different individuals in response to the same macronutrient stimuli. And that's the part of the talk I'm super excited about because it is my project and what I'm currently working on. So I'm hoping to share with you some very preliminary data that hasn't been published elsewhere yet. Okay, so before we begin, we need to be able to understand what the difference is between a simple correlation and an actual causation. So just because event A and B are occurring at the same time, does this now mean that A is causing B or B is causing A? We don't know. So how do scientists determine what is causal amongst multiple things that are happening at the same time or correlated with each other? So here I'm going to use a very silly example to show you what the difference is between a simple correlation and a direct causation. So here we have a very cute cat and we have an office chair. And the scientific question we're going to be asking is, how does this office chair stay upright? So for the next half hour, we're going to pretend we don't know how an office chair stays upright. But what we do know is that when the cat and the chair are in the same room together, the cat really likes to sit on this chair when it's in an upright position. But the cat has absolutely no interest in the chair when it's sideways. So does this now mean that the cat is keeping the chair upright? Of course, we know that's not the answer, but how do we prove this scientifically? So the first thing we do is we take the cat out of the equation. And lo and behold, the chair is still upright. So of course, the cat is not keeping the chair up. But what is keeping the chair up? So scientists, we like to do things called loss of function experiments. So we're going to remove certain parts of the chair, one at a time, to see which component of this chair is keeping it upright. So of course, one of the first things we're going to remove is the legs of the chair to see what happens. And we all know when we remove the legs of the chair is no longer upright. So this is now a direct causation. The legs of the chair are required for the chair to stay upright. It's not the cat sitting on the chair, which is just a simple correlation. So what are some correlations that we know that are relevant to insulin and diabetes and obesity? So we know that excess weight gain, or obesity, is associated with elevated insulin levels, also known as hyperinsulinemia. So we have too much insulin in our bloodstream. We also know that insulin resistance is also associated with hyperinsulinemia. Now briefly, insulin resistance is a state by which your insulin no longer works properly. So your tissues that are responsible for taking up glucose from the bloodstream are no longer responding to this insulin. And this results in more insulin being released. And that's insulin resistance and hyperinsulinemia. So this is the standard obesity insulin diabetes model. So an unhealthy diet will lead to weight gain and eventually obesity. And this will lead to insulin resistance, so that state at which your tissues are no longer able to uptake glucose from the bloodstream efficiently. And this results in hyperinsulinemia. So now, because your tissues are no longer responding to the insulin as well, your beta cells, which secrete insulin, are now going to work really, really hard to release more insulin to compensate for this lack of the effect of insulin. So now you become hyperinsulinemic. However, eventually, your beta cells are working really, really hard to deal with this insulin-resistant state, and they're releasing so much insulin that eventually they become exhausted. And once your beta cells become exhausted and they're no longer able to keep up with the demand for insulin, this is when we start seeing type 2 diabetes. So that's the consensus model. It's quite straightforward, it's quite linear. However, we all know that things are not always as they seem. And so our lab, data from our lab, as well as data from a number of other groups, has suggested that this is more of a circular model as opposed to a direct linear model. So a lot of data shows that too much insulin is actually contributing to not only obesity, but an insulin-resistant state as well. So when we're talking about insulin here contributing to obesity, we're talking about the state of hyperinsulinemia, so too much insulin. Of course, we all know that insulin is actually good for us, and we need it to be able to control our blood glucose levels. But when we're talking about insulin contributing to obesity here, we're talking about excess insulin. So if excess insulin is contributing to obesity and insulin resistance, how do we go about proving this as scientists? So if you remember, I talked about those loss of function experiments where we try and remove something to see what happens. So of course we can't totally remove insulin because either the animal or the patient would die, but we can try and reduce insulin levels to a certain amount where the patient or the animal is still maintaining their blood glucose levels. And so a number of groups have shown that insulin indeed does contribute to weight gain and insulin resistance. For example, patients that are on long-term insulin therapy do tend to gain weight compared to patients that are not on long-term insulin therapy. And in patients who are obese and hyperinsulinemic but not type 2 diabetic yet, if you give them a drug that blocks some of the insulin they normally release, they actually tend to lose weight. And in popular culture, there's been a number of books that have been published that deal with this state of hyperinsulinemia. And so these books focus on the main stimulator of insulin release, and that is, of course, carbohydrates or glucose or sugar. And what they say is that too much sugar will lead to a state of hyperinsulinemia, which then contributes to obesity and insulin resistance. Okay, so how do we go on about proving that too much insulin is not good for us? So of course we can't do these experiments on humans. You can't bioengineer a human subject. So we move on to the next best thing, and we use a mouse model. And so the question is, can we engineer mice to test whether too much insulin plays a causal role in weight gain, or is this simply just a correlation? So I'm going to present to you some data from our lab that they've obtained this over the past 5 years or so. And what they did is generate a mouse model where they knocked out or removed one copy of the insulin gene. So we all have 2 copies of every gene, just like insulin. And what they did here with this mouse is they removed one copy of that insulin gene. So that mouse was still produced insulin, but it produced less insulin. And that mouse was also able to control its blood glucose levels, so it did not become diabetic. But what's special about this mouse is if you place this mouse on a high-fat diet, this mouse does not become hyperinsulinemic. So if you take control mice, so mice that are not bioengineered, and you put them on a high-fat diet, that's in the light pink here, these mice eventually become hyperinsulinemic over time. So they start producing a lot more insulin. However, if you take these bioengineered mice, so the mice who were removed one copy of the insulin gene, they do not become hyperinsulinemic. And what's interesting about these mice is even though they are producing less insulin, they're still able to control their blood glucose levels, so they do not become diabetic. And they're also still quite sensitive to the insulin. If anything, they're actually even more sensitive to the insulin than their wild-type controls. So now what we have is this mouse model that we prevent it from becoming hyperinsulinemic, but it's still able to control its blood glucose efficiently. What happens to body weight in this mouse when you place them on a high-fat diet? So I apologize for the graphs look a little small, but I'm just going to take you through what they mean. Basically what we're saying here is this mouse model where the mouse does not release as much insulin. When you place them on a high-fat diet, they do not get obese. So even though they're getting the same diet as their wild-type counterparts, when you take away that one copy of the insulin gene, the mice do not become hyperinsulinemic and do not gain as much weight as the control mice when placed in a high-fat diet. And this is a direct cause of less fat in their fat pads, as opposed to them losing weight in their skeletal muscle or any of the other organs. So basically, they're leaner mice, even though they're eating the same thing as their other control mice friends. And as you can see here, so this is a biopsy from their fat tissue or adipose tissue. And the control mice, when placed on a fat diet, high-fat diet, their adipocytes expand and swell. However, the mice that have been bioengineered and lack that one copy of their insulin receptor, of their insulin gene, they do not have that same expansion of their adipocytes. So these mice are leaner. So the next question is, does this modest reduction in insulin have a long-lasting effect on weight gain? How long does this last? So here we have a very similar but slightly different mouse model where, again, we delete that one copy of the insulin gene. However, these mice are a little bit different where they do not become hyperinsulinemic throughout the majority of their life. However, towards the end of the life, they do become hyperinsulinemic, just like their control mouse counterparts. However, when we look at body weight, these mice still remain leaner than their control counterparts. So what we see here is that if you reduce insulin levels in these mice when they're young, they still maintain that protection from becoming obese. And what about insulin sensitivity? So we talked about that as a measure of how well the tissues of your body are responding to insulin and how well they're taking up glucose from the bloodstream. And what we actually see in these mice that do not become hyperinsulinemic is that they're actually better able to uptake glucose from the bloodstream. So their insulin, they have less insulin, but the insulin works better. And very, very exciting and very interesting is that these mice, they live longer. So there's about a 10% increase in their lifespan when you reduce insulin levels by about 10%. So we thought that was really exciting, that you're able to expand the lifespan of these mice by about 10%, which is a lot for a mouse that only lives about 2 years. Okay, so what about therapy? So what if we first make these mice obese, so they gain weight, can we now induce them to lose weight by removing one of those copies of the insulin gene? And so that's what we did. We placed normal mice on a high-fat diet. They become obese. And then we try and remove that one copy of the insulin gene after they become obese and see what happens. And what we do see is that these mice, again, have less insulin. Less insulin is being released, but they're still not diabetic. They're able to control their blood glucose levels really well, but they lose weight. So they become leaner than the mice that still have both copies of their insulin gene. And these mice both groups are eating the exact same thing. They're not moving more. Nothing is different apart from the fact that they have less insulin. Okay, and so for conclusion of the first part of the talk today, what I've shown you is that reducing insulin levels prevents diet-induced obesity in mice. And reducing insulin levels just for a short period of time while these mice are young provides long-lasting, sustained effects on these mice not becoming obese. Insulin reduction also improves insulin sensitivity, so your body is better able to use the insulin it has. And these mice tend to live longer. And we are able to induce weight loss in mice that are already obese once we remove some of that insulin. Okay, so remember I told you that the original model stated that unhealthy diet leads to obesity, leads to insulin resistance and hyperinsulinemia, or too much insulin. The new model actually suggests that an unhealthy diet will first lead to hyperinsulinemia. So it will lead to too much insulin being released from your beta cells, and that in turn will contribute to some of the weight gain we see, and then insulin resistance and beta cell exhaustion, and eventually type 2 diabetes. So what we're trying to say is that this pathway is a lot more complex and is actually a little more circular than linear. Okay, so that was the first part of the talk. In the second part of the talk, we're going to be talking about how insulin secretion may be different between different individuals. And again, this is something I'm really excited about. I've been working on it for about 6 months now, and I'm going to be showing you a little bit of preliminary data, but in about a year or so, I hope to have a lot more data to show you. Okay, so maybe I should have had this slide earlier, but just to kind of get us all on the same page, we need to know where insulin is actually released from. I'm sure we all know that the pancreas releases insulin, but which part of the pancreas? So the pancreas actually has two specific functions. It has an exocrine function and an endocrine function. So the exocrine function is actually the majority of the pancreas, and that's when you eat a meal, the pancreas will release a bunch of enzymes that will help digest the meal you have just eaten. But small parts of the pancreas form the endocrine tissue, and those are these tiny little red balls you see here. So each one of these tiny little balls is called an islet, and each islet is composed of about 1,000 cells. And there's different cell types within the islet. So there's alpha cells, beta cells, delta cells. They all have a different function. It's the beta cells within the little islet that actually secrete insulin. So it's the beta cells within the islet that's found in the pancreas that releases insulin into our bloodstream. Okay, so this is actually quite a complex process. It's not as simple as people think. A number of different channels and processes are involved to actually release insulin from each beta cell. And insulin is not just kind of found dispersed throughout the beta cell. It's actually stored. And it's stored within these tiny little secretory granules. And it takes a lot of events to occur for one of these granules to be released from the beta cell. And most people think that glucose, or sugar, is the only thing that will release insulin from the beta cell. And whilst glucose is the main stimulator, fats and proteins will also potentiate insulin release. So they will also release insulin from the beta cell. However, it seems as though fats, proteins, and glucose or carbohydrates, whilst they all release insulin from the beta cell, it seems like different individuals respond differently to different macronutrients. So not everyone will respond the same way to the same load of glucose or the same amount of protein or the same amount of fat. So we are very privileged and very fortunate to receive human tissue that has been donated by patients that have been deceased, and they donate their organs for research. And so we're very fortunate and privileged to receive some of these organs. And one of the organs we do receive is the pancreas. And so we're able to look at the specific islets from different individuals and see how they will respond to a certain amount of glucose, a certain amount of fat, a certain amount of protein. And so that's a project that I'm currently working on. So I'm just going to take you through what we're currently looking at. So these are islets from one specific individual. We're going to call them individual X. And what we're looking at here is insulin secretion from individual X in response to glucose, protein, or fat. And so this individual, as you can see here, releases far more insulin in response to protein than they do in response to a glucose stimulus, which is actually quite unusual. We think that most people will respond, will release more insulin in response to glucose, but this individual releases more insulin in response to a protein stimulus. This individual does not at all release any insulin when given a fat stimulus, which is also quite interesting. And if anything, when giving a fat stimulus together with a glucose stimulus, this individual is releasing less insulin. So what we're trying to look at is a number of different donors. So we're trying to get a giant database of islets from different individuals and see how each one of those islet preparations will respond to a different concentration of glucose or protein or fat. And again, what we're seeing is that not everyone is responding the same way. So for example, individual D here, very little response, very little insulin being released in response to a glucose stimulus. Compared to individual B, for example, their islets are quite responsive to glucose. However, when we look at protein, they're quite similar in their response to their protein. And then we look at the fat stimulus. Individual B does not respond at all, so no insulin is being released in response to fat. However, when we look at individual D releases quite a bit of insulin in response to a fat stimulus. Because as you can appreciate, so not everyone will respond the same way to the same meal. So you may give somebody a steak, and they might release quite a bit of insulin, versus another individual might eat a steak and they're not releasing very much insulin. So the question now is why? Why do different individuals respond so differently to the same macronutrient stimulus? And so a number of groups have actually tried to understand this. They did not look at fat or proteins, they only looked at glucose. But what they did try and see is, does someone's BMI correlate to how they will respond to a glucose challenge? Or does someone's age correlate to how they will respond to a different glucose challenge? And they were not able to find any direct causations here. So somebody who has a BMI of over 30 looks like their islets respond just as well to glucose as somebody who has a BMI of under 25. Now these are non-diabetic individuals, of course. Another group looked at genes. So do our genetics play a role in how our islets function? And so again, this group just looked at the response of the islets to glucose. And what they saw was that there's quite a bit of a substantial difference in a number of key genes that are involved in islet function. So it looks like islets that respond to glucose really well tend to have more of the key genes involved in islet function than the islets that do not respond to glucose very well. So the big question is are we able to use genetic profiling to understand the mechanisms underlying these variations in insulin release between specific individuals? And unfortunately, we don't know yet, but that is going to be my project, and we really do think and hope so that we can do this. And as you can see, the end goal of all of this is sort of to be able to personalize someone's nutrition. So to tell them, okay, your body will release a lot of insulin if you eat this meal, but if you eat this other meal, you will not be releasing that much insulin. So again, in conclusion for the second part of this talk today, what we know is that islets from different individuals have a varied response to a different macronutrient stimuli. So not everyone responds the same way to a glucose challenge or a protein challenge. And data suggests that these differences are not due to gross characteristics such as age, sex, or BMI. And it looks like that we can probably use some sort of genetic profiling to be able to understand how an individual will respond to a specific dietary challenge. Okay, so I'd like to thank you for your attention and thank my colleagues at the UBC Diabetes Research Group and the Institutes for Personalized Nutrition for allowing me to present their data. And I'd like to thank the CIHR and the CDA for funding this work. And I would be happy to try and answer any of your questions. So thank you.
Thank you.
Thanks.
Lots of thanks going around. And we do have time for a couple of questions. If
Sure.
you're open to
Yeah,
that.
of course.
Fantastic. And one right up front.
Have you been exploring the gene profiling in relation to the microbiome?
That's a very good question. Unfortunately, we haven't done that yet. So the islets or the donor tissue we get, it's only the islets. So we don't have any information about their gut microbiome. But it's something we can probably look at in a mouse model to see if the gut microbiome does affect their islet function. So, yeah. Sorry?
Like gene expression?
Yes, of course. Yeah, gene expression, yep.
Incredible how much we can learn from our little friends, hey? Do we have any other questions? Anyone? Perfect. Just gonna shimmy on down. Sorry.
I was just wondering about diseases that can make you diabetic. Is there, I know, something that would be helpful with those? Like
Studying diseases that contribute to diabetes?
yeah, is there anything that you would suggest for something like if you got, I can't even think of one, but but there is some illnesses or diseases out there
Of
that
course. Yeah.
are prerequisites before getting diabetes.
Yes,
You don't
yeah.
just get straight diabetes.
Correct. No, there's
Yeah.
there's quite a few things that do contribute to the development of diabetes. Unfortunately, it's quite complex. I can't tell you specific diseases that can be contributing to this, but it's something that we can look at. So the islets we do get limited information about the donor, but we can get information on whether they were suffering from something prior to the diabetes. So it's something to look into. Thank you.
Awesome. Thank you for the question. One more.
Yes. Sorry, I was just also wondering, did you put any of these mouse groups or individuals
Yeah.
or whatever, what type of medication did you give them, or or was it just straight
So they
stimulant?
were not on so these mice.
Yeah.
The special thing about these mice is they're not diabetic, so they do not need any medication. So yeah.
And we have time for one more question. If we have anybody, perfect.
For diabetes type 2, it's better take medicine or on dieting is better.
Or
On diet.
It's definitely, if you can control type 2 diabetes and control your blood glucose levels without medication, that is great. And it's better to try and control your diet for sure. But unfortunately, there are a number of people who are unable to control their diabetes that way. So
All right, well, I think that wraps it up for us. It's already, I think it's almost
Thank
2.
you.
This is incredible. So please, everybody, put your hands together one more time for Dr. Jelena Kolic.
thanks.
You've been wonderful. Thank you so much.
Thank you.